Once the alignment step is complete, we can go ahead and click on Continue. The web server collects information from all the quantified results and combines them into a matrix of gene level counts, which has been previewed below. We can now click on Continue and proceed to the next step of notebook generation. Here, the user is required to input metadata describing the samples in the uploaded dataset. The user can either specify the group the samples belong to, using the form on the left, or upload a sample metadata file using the form on the right. So in this case, we'll click here and select the sample metadata file from the file system. In this case, we can then see that the first 3 samples in the dataset correspond to a wild-type group, and the remaining 3 samples belong to a knockdown group. We can click on Continue. And during this process, the data and metadata are packaged into an HDF5 file and uploaded to the cloud, where they're made available for notebook generation. The user is required to select 1 or more computational tools which can analyze the RNA-seq data. And BioJupies currently provides access to a library of 14 plugins which are divided into 4 categories: exploratory data analysis, differential expression analysis, enrichment analysis, and small molecule queries. The user can add or remove each of these tools by clicking on the buttons on the right. An option will reveal more information about each tool, including a brief description, links to references, interactive examples, video tutorials, and the source code hosted on GitHub. So we'll select a couple tools from each of the sections. And then click on Continue to proceed to the next step of notebook generation. The user is required to specify 2 groups of samples they wish to compare in order to generate a gene expression signature. So first of all, the user is required to specify the names of the groups. So we'll compare the wild-type group to the knockdown group. BioJupies automatically predicts the groups based on the names, but the user can also optionally toggle each group using the drop-down menus shown here. Once the desired groups have been selected, the user can then click on Continue and proceed to the final step of notebook generation. The user can optionally customize the notebook by changing the notebook's title, adding tags which specify the tissue or cell line the samples belong to. In this case, these match different ontology terms, such as the Uber anatomy ontology or the cell line ontology, as well as information about the diseases, drugs, small molecules, or gene perturbations the samples have been treated with. Optionally, the user can modify parameters for each of the tools. In this case, for PCA, modify the normalization technique, whether to apply z-score, and the amount of genes to be used for calculation of the principal components. Once the user has selected the desired settings, they can then click on Generate Notebook to launch the notebook generation job. Once the process is complete, a link to the notebook is displayed on the website, alongside some options to share it on social media, and a summary of the signature and the analysis tools used in the notebook.